So I get to talk with you about my favorite topic. Oh no, I get to talk with you about biotech and food. Okay, I am Tom Zinnen. I have to do my own introduction. I was born in Dixon, Illinois. Went to high school at Dixon, Illinois High School. Went to UW-Platteville and studied biology there. Biology is the study of bile. I know. Then I returned to the fair state of Illinois to go to the University of Illinois, got my master's degree there in plant pathology. And I came here on January 7th of 1982. It was 20 below zero. And got a PhD in plant pathology working on plant viruses. Then I went to did a postdoc on the east side of Madison at Genetics, which is no longer there. I killed it. Then I spent 6 years at the Northern Illinois University Plant Molecular Biology Center. Then on Monday, June 3rd of 1991, I returned to UW-Madison for the job I currently have. I've had 2 little stints in Washington in 2000, 2001 as a Congressional Science Fellow. I worked on the majority staff of the House Committee on Agriculture. The majority was controlled by the Republicans, and the committee was chaired by the gentleman from Lubbock, Texas.
Who's that?
And it still makes me, it doesn't matter. It still makes me smile to think about them hiring a guy from Madison, Wisconsin. Because the folks in Lubbock think there's nothing but a bunch of damn liberals over there in College Station at A&M. Then I also spent 20 months writing speeches for the director of the National Science Foundation from 2008 to 2010. As you can tell, I've willingly and delightedly returned to this place once, twice, thrice. It's a great place to be. Thanks for coming tonight. We get to talk about biotech and food, dilemmas in communication and culture. Oh. You've seen this before if you've heard one of my talks. I came here as the bovine growth hormone inspired and funded state specialist in biotechnology. And it doesn't take very many moments in Wisconsin before you realize, especially in this state and especially in dairy, that biotech and food is a very profound issue. It's a big part of our culture. So some of the things that I enjoy most about this job as an extension person is getting people to ask, what is science? How is science different from other ways of knowing? To get them to talk about something that I never had a class in, but it's called Epistemology. It is that branch of philosophy that asks, what is knowable? What is the nature of proof and disproof? How do we know that? How do we show that? How do we test that? How do we know it's not someone else? Epistemology is the branch that asks what is knowable. Ethics is the branch that asks what is noble. One syllable difference. About 20 years ago, people decided that we should combine math, engineering, technology, and science and call it METS. Very good. That was very good. 1969, I know. Well, you could say you're sorry, but that's okay. You moved. Then somebody had the brilliant idea, well, we shouldn't call it METS, we should call it Science, Math, Engineering, and Technology, which is SMET. Some botanist got a hold of said, let's call it STEM. And I still think this is one of the big questions in public policy and how science bears on how we make personal choices and form public policies is how we look at epistemology, particularly how do we look at science, technology, engineering, and math, and how we think we know what we know and how we make decisions in the face of uncertainty. So here are some other ways of knowing. These are all very fond. What do you pay to go to college? A lot. Way to go. Reason, logic, math, intuition, instinct, tradition. That's the TEVI one. Authority. Empiricism is my favorite. Experimentation especially. strong inference, history, literature, revelation, prophecy, mysticism, mythology, experience, superstition, imagination, naive theories. Those are all the things that are, at least, and probably more, that are running around in all of our heads and in all of our communities, competing for attention and persuasion. So the great thing about studying biology is that it's also the study of food, because we are obligate chemoheterotrophs. That means we got to eat other things that have been alive, plants, animals, microbes. Starting approximately 10,000 years ago, is anybody there to report? No. Humans started making these things into crops, livestock, and cultures. And what were we selecting those things on? Taste, color, ease of preparation, yield, vigor, nutrition. Did I mention taste? So I'm not a food scientist, and I no longer play one on television, but there are two dirty little secrets, at least, of the food industry. And one of them is, the dirty little secret of the food industry is taste. And we'll get to the other one. This is very interesting because many people, especially in plant breeding today, and if you've read the newspaper articles coming out of the College of Ag and Life Sciences in the last week, you'll realize that there has been a lot more attention in the last 20 years, 10 years, especially the last 5 years, in breeding for taste. Because so much taste was bred out of cultivated varieties of crops while they were breeding for other things like disease resistance or yield or shippability. What is a trait? What is a traitor? A trait is when you have a combination of the genes by environment. That means if you want to change what the traits look like, you've got three possibilities. Change the genes, change the environment, change the genes and the environment. Farmers, growers do all three. And keep in mind that we have these things called genes. Most genes are encoded by DNA, and genes flow in nature, within species, between species. Transformation, transduction, conjugation, cell fusion, viral infection are just some of the words. I'm particularly fond of some of these because a gentleman by the name of Joshua Lutterberg, one who worked across the street here from 1949 to 1958, won a Nobel Prize for discovering some of those methods of gene flow. DNA is not the only carrier of genes, but it's the one we spend the most time on. And for our purposes with genetic engineering of crops, there's a very big one called Agrobacterium tumefaciens. Causes this disease of plants called crown gall, and it's a pioneering tool in biotech. One of the things I promised in my little missive is that I would talk about aspects of research done at UW-Madison. There's a gentleman by the name of A.J. Riker, who was a plant pathology professor here back in the 30s and 40s, and he worked on this disease. And he tried to do a very interesting thing, and that was he tried to frame it in his messaging as plant cancer. Where do you think he might be able to get research money from? If he could frame it as a model of cancer. So agro means field, bacterium means bacterium, tuma means tumor, and faciens means making. So this is the tumor making bacterium in the field. And you spread it by hoeing. And when you hoe, what do you do sometimes? You whack the stem of the plant where it meets the soil. And of course, as our horticultural expert will say, that's called the crown of the plant. Why? I don't know. Botanist. Okay. Hey. So here's some pretty cool stuff that I need to touch on to get to other stuff, and that is I don't know, what's the math on 72 years? 45 years ago? This gentleman, Paul Berg, did some synthesis, was able to put chunks of DNA from two different sources together, and they were biologically active. He did not use the same cut and splice technology that we used, that Cohen and Boyer used in 73, but he kind of led the way for them, and I think all three of them ended up winning the Nobel Prize in Medicine or Physiology. What does this mean to have recombinant DNA technology? What we're going to be talking about now, and what's in contrast to gene editing, which we'll talk about in a little bit, is, wow, if you're a plant breeder, what did your gene pool do? Yep. 1970, if you're a corn breeder, what's your gene pool? Corn and its close allies. 1973, at least theoretically, what's your gene pool? Anything that's alive and has DNA in it. And because of work that Howard Temin and others did here and at other places, it doesn't necessarily have to be solely DNA, because what can you do with RNA? You can reverse transcribe it and make it into DNA. And I worked on a virus called tobacco mosaic virus, which does not have DNA in it. It has RNA. But can you reverse transcribe that and then stick the DNA that you make into a plant? Yes, you can. And so that's the second Nobel Prize, one for research done on this university in medicine and physiology, that I want to make sure people are aware of, this kind of background stuff. So, any organism on Earth is a source for genes for use by plant breeders. Since we're talking about communication, this is something, I don't know, I always thought this was true, and it never sat right with me when I'd hear my colleagues say, oh, this is just another tool. It's like, well, that might be true, but it's kind of like saying a sawzall is just another saw. Careful where you point that thing. I think going back at least as far as Aristotle and other people that have done formal studies of rhetoric and the art of persuasion, sometimes you're better off just saying, whoa, it's really this way. It's really powerful. Because that allows you to go to the next stage and saying, just because it's really powerful, does that mean it's more dangerous? Because power and danger may or may not be linked. So here's some pretty cool things to think about. In 1977, whoa, we can move. Not only does agrobacterium move genes into plants on its own, people started figuring out that they could do it in the lab, and therefore agrobacterium could be a lot like a vector. A vector is another word for envelope. You write a letter. Do you put the letter in the mail? Well, you can. What do? You put it in an envelope. And what do you put on the envelope? The address. And then you put a stamp in it. And then what happens? Whatever's inside that envelope gets delivered to? Yeah. And that's pretty cool. That's what a vector is. That's a reasonable analogy for what's going on here. Agrobacterium has DNA that it inserts into the cells of plants, and it's not just into the cells of plants, it's into the DNA of the plants. So if you have agrobacterium DNA here and here, what happens if you go like this and put some of your DNA in between? Can you stick two letters into one envelope? Yes, you can. In 1983, there was the first expression of a protein of a transgene moved from a bacterium to a plant. This is petunia and tobacco cells. And also in 1983, done on the 6th floor of Russell Labs, and other floors there, but that was the primary floor. First expression of a protein of a transgene moved from a plant to another plant. So the first one, which has primacy, was Mary Dell Chilton's group, and she moved a bacterial protein, a gene for bacterial protein, into a plant cell and got it to express. The contribution from researchers here at UW-Madison and these are the ones that also started agrogenetics, is the idea of, wow, can you move a gene from a plant, one species, into another species and get that gene from the bean to talk to the sunflower and be expressed in the sunflower? And the answer was yes. And so they called this the sunbean. Now remember, when you want to communicate something, you want to make stuff memorable. I like that. Question?
Is it possible to turn up the mic a little bit?
Yes, it is. It's even possible for me to talk louder. Just don't let my wife know. Okay, I'll try to do that. Oh, she's calling. She would like to FaceTime. Sorry about that. Yeah, it's bad. Okay. All right. Holy cow. Same year, but not published that year. 1983, Carey Mullis figured out the polymerase chain reaction. That is pretty cool. And that's the idea that, whoa, you can take and amplify a chunk of DNA or RNA if you first do a reverse transcription. And by going back and forth, kind of like what she's doing here, knitting. Have you ever seen one of those knitting machines that go back and forth? That's a lot like what PCR is like. Except for PCR works by doing something at 37 degrees centigrade, human body temperature. And then to get the DNA to melt, what do? You heat it up to? Okay, 92. It's my favorite radio station. I don't know. But pretty hot, you know. What does it do to the polymerase? It denatures it. So what do you got to do? Every time you do a cycle, you got to add more protein back, add more of the polymerase back. Except, holy cow, what happened? Some pin-headed academic from the Indiana University took his students out to Yellowstone National Park. And what is it, Yellowstone National Park? Hot springs. And are they colorful hot springs? And if you're a geologist, they're very colorful because of the minerals. And if you're a biologist, they're very colorful because of the bile. Okay? So in the mid-60s, Thomas Brock and his crew, I think, is going out there, and they come back with this bacterium that they characterize and call a new species, and it's called Thermus Aquaticus, which is very complex Latin for hot water. Okay? Now, if you're living at 93 or 95 or 98 degrees centigrade, what does that mean about your proteins? They like it. So I don't remember who came up with this. Oh, by the way, Thomas Brock, very quickly after that discovery, moved from Bloomington, Indiana to Madison, Wisconsin, and is now a professor emeritus here. So I don't know the whole story of Thermus Aquaticus polymerase, but if you've ever heard of TAC polymerase, TAQ, that came from that discovery, totally out of the blue, unintended. And one of the major producers of TAC polymerase is Promega. Okay? And if you ever want to read a fascinating story of patent interference, you can either read the entire history of Whorf, or you can go read about the TAC polymerase patents that Promega successfully prosecuted. Holy cow, now we got plants, we want to test them out in the field. And that was a big deal. I don't know if you remember some of the rogue people that put stuff out without being approved. Yeah, it's always helpful when people go out on their own. But many people don't realize where was the first approved release, field test, whatever you want to call it, of a genetically engineered plant in North America? In Middleton, Wisconsin. Now, why was it in Middleton, Wisconsin? Because Verona wouldn't have it. Because that's where another company, not Agra Genetics, but Agra Cetus was located. And if you would ever like to see a tale of two cities or a tale of two corporate cultures, comparing Agra Genetics and Agra Cetus is a very enlightening thing. I forgot to mention today that I promised that I would use the 2.5 happiest words in the English language. After this talk and the Q&A, we will adjourn to the terrace and I'll buy. And I'd be happy to talk to you about the difference in the cultures of those two places, at least in the era of 1985 and 1986 when I was at Agra Genetics. Okay. Now, the first approved field test of a transgenic plant, what kind of plant was it? Tobacco, not tomato. Why tobacco? Okay, what did we love to grow here in Wisconsin? Tobacco. Does tobacco get diseases? Yes. Do we have a department of plant pathology? Yes. What's that? Holy cow. Have I got stories for you? Okay. So people don't realize this. If you go over and look at the Curry murals in biochemistry, which are one of the major reasons why that building was not torn down, the fact that it was home to the discovery of vitamin B and how to make vitamin D and warfarin and a whole bunch of other stuff helped too. Over on one of the panels is a painting of crops. And the crops that you can identify are corn and potatoes and tobacco. You're from down by Oregon, right?
Yeah,
Stoughton, even more so. So Viroqua, Stoughton, and Edgerton were the big three towns. Tobacco gets a thing called tobacco mosaic virus. That's what I ended up doing my PhD research on. Tobacco mosaic virus was one of the first viruses ever purified. It was the source of where people figured out, wow, you don't have to have DNA because what kind of nucleic acid is in TMV? RNA. And a gentleman who worked on this campus in the 30s came this close to being the first person to purify a virus. And that virus was tobacco mosaic virus. A couple other people worked on tobacco. Well, a lot of people worked on tobacco as a model organism. And the two that I want to mention by name are Murashige and Skoog, because Murashige, as part of his research here, came up with the medium that has the right kind of plant hormones. God, I was relieved to find out plants have hormones. And that's how you can, one way that you can grow plants from cells to callus to shooting and rooting medium. Okay, so that's another major contribution in this whole area from folks who worked here. Because of that, tobacco was a huge model organism. It was one of the first, if not the first, well, either petunia or tobacco, and they're both in Solanaceae. Tobacco is widely used as the little white rat of plant molecular biology, and that's why what went on in the field was transgenic tobacco. And the consultant that they hired was a gentleman by the name of Robert Fulton, who was my major professor, and he's the guy that taught me how to grow and rub tobacco. It's a great thing, rubbing tobacco. Okay.
What did you put in that tobacco, that first one? What was transgenic about it?
Oh, you want a straight answer. Darn. I can't remember. I don't know if it was just a selectable marker or something that was easily scorable. I don't remember. At that time, it didn't matter if it was transgenic. You know, you had to jump through all kinds of hoops. Now, in 1986, automatic automated DNA sequencing was also developed. This was developed at Caltech by a guy by the name of Leroy Hood and his associate by the name of this is embarrassing because I can't pull his name up right now. He's the associate director of the Biotech Center. It'll come to me. Remember, I turned 60. Get out of jail free card. Now, the other thing that happened in 1986, I don't know if I have it here, that's important, is that was also the year that they got approval to do the frostband field test. What is frostband? It's a ice nucleating bacterium. Folks here discovered in the Department of Plant Pathology that, hey, one of the ways that, what do we used to seed clouds with? We drop little kernels of corn from 20,000 feet. No, what do you do? Some sort of potassium iodide or something like that? Something like that. Well, what people also figured out is, wow, bacteria can nucleate ice. And if you nucleate ice, what do? You coalesce raindrops. And if you want to if you want to be a rainmaker, there are two ways to do that. And the biological way is through ice nucleating bacteria. However, you may not want ice nucleating bacteria on your crops when the temperature gets down around 33, 32, 31, because what will it do? It makes it freeze. Instead of the water being super cooled, it'll freeze, and then boom, you get frost damage. So that was all work that was developed here, and then the researcher got his PhD and went out to UC Berkeley, and he helped start a company, and that's where frostband came through. It was field tested in 86 or 87. The coordinated framework for regulation biotech is something we'll be talking about a lot. There's the first frostband. That was the first approved test. Recombinant DNA to chymosin. What's it take to make cheese? A master cheesemaker. Takes milk, takes salt, takes bacteria, and enzyme. And what's the name of the enzyme? Rennet. And rennet has a specific protein in it called chymosin, which is a Yiddish word for stomach slime. Actually, it's a Greek word for stomach slime. Okay, but it's a protein, which means it has a DNA chunk that encodes for it, and that chunk of DNA is found in cattle cells. What if you could take the cattle cells, find the chunk of DNA, copy it, move it into bacteria, grow up a little bacteria that now have one extra gene in them, and if everything went right, what could that bacterium make? Chymosin. And then you could purify the chymosin out, and what could you do? You could have a source of chymosin independent of the fluctuations of the dairy veal industry. You could also talk to your local rabbi and local imam to see if, wow, if you make the bacteria, if you make the chymosin in a bacterium, does that bypass milkish and fleischish? Could you have kosher cheese, cheddar cheese? That's not answerable by scientists. You can go ask a rabbi. And since rabbis are a lot like Catholic priests, well, not really, but I was raised Catholic. What do you do when you don't get the answer that you want from your priest? Go find another priest. So, ask my parents why they have their seventh child. So, you don't know what the answer is going to be, but if they arrive at an answer, at least for whoever follows the rabbi or that organization's kosher, you can say, this is okay. I have a 1-gallon jug of chymosin from 1990. It's still in the refrigerator. It is still active. It's recombinant. How controversial was chymosin? 0. Where was one place that was heavily studied? 1.5 blocks west of here at Babcock Hall. Holy cow, 1992 USDA Food and Drug Administration declares, oh, that's extra. This should not be there. Biotech-derived foods don't differ in any uniform ways from foods derived from other stuff. That's a big deal because, 1993, we now have bovine growth hormone approved on November 1. Did it actually get approved? It got approved, and then it got delayed. Why? By whom? Who was the senator from Wisconsin at the time?
Feingold.
Russ Feingold. How did Russ Feingold make his name?
BGH.
One of the ways is through opposing bovine growth hormone. Where did Russ Feingold live? Middleton. Oh. Okay. Huh. I like that. Huh. Okay. How many Democrats were in the Senate at the time? 60. Sorry. Yeah, I know, it's kind of hard to imagine. Okay. How many did you need to overcome filibuster? 60. So, wow, what did that make every Democratic senator? So it was possible for a single senator to get into the appropriations bill rider that would say, well, this isn't going to go on November 1st. We're going to have a 90-day delay so we can study this further. Okay, so that's another way that Wisconsin has played a significant role in this stuff. 1994, just right after BGH became legal after the 90-day hiatus, flavor saver tomato. 1995, transgenic field crops were approved, specifically soybean and then corn. And so 96 was the first summer that they were planted in the United States. Dolly the sheep was born on the 5th of July, but you didn't hear about it till the following January or February. Okay? And did it put everybody in a tizzy? Yes, this was 19 years after who was born? 18 years after who was born? Louise Brown. And who was she? First test tube baby. So remember when she was born and everything was going to HE double toothpicks? So when I started this job, I couldn't draw on a whole lot of biotech things because it was all happening under our feet. But I could ask people, how many people in here, I'd have a room of folks, I'd say, how many people in here have a child or grandchild born from in vitro fertilization? And then I'd say, how many people in here know somebody who has a child or grandchild born from in vitro fertilization? And then I'd say, how many of you want to have that technology taken away? Because human IVF, I think, is one of the great examples of shifting from sacrilege to sacrament. When it first came out, everybody was against it, except for people who wanted to have a baby. Not very many things do a significant change like that, from sacrilege to sacrament, in under 20 years. Then we had the transgenic papaya, we had potatoes boycotted, and golden rice announced. Here's the second dirty little secret of food science. Always, always, always protect your brand name. Always, always, always protect your brand name. So who's more vulnerable, Monsanto or McDonald's? They both begin with M. Monsanto is not a food company, and McDonald's is. So starting in 1997 and 1998, the folks who were against this got very smart because they stopped trying to boycott Monsanto and they started boycotting Nestle, McDonald's, all these folks that have to follow that big rule of food companies. Then through the grace of the deity, Starlink corn contaminated the single stream of U.S. corn. This was in November of 2000. That's when I was trying to get a position on Capitol Hill, and gee, suddenly I had useful talents. That was also the year that the Arabidopsis genome was sequenced, and this university is a major center, or has been, in this plant called Arabidopsis. And my boss in particular spent a couple of decades working on Arabidopsis. Now we're going to skip ahead. There's nothing really happened in the 20s. Here, this thing called CRISPR comes in. There had been other gene editing tools. They were kind of klutzy and clumsy. You could see the power of gene editing, but people couldn't figure out how to get them to fly easily. And once they figured out, Jennifer Doudna and Emmanuel Charpentier figured out how to combine the Cas9 protein with the CRISPR. Now you got this cool gene editing tool. I don't know if you ever remember, but I remember the first time I ever edited a document on a Wang computer. They had to pick me up off the floor. And even more astonishing, in 2014, the USDA starts giving go-ahead to gene-edited crops. Why? The whole framework for the U.S. way of regulating transgenic plants was it had to be a transgene. In other words, the whole gene had to be transferred. What happens when you genetically engineer without adding something? No transgene. No transgene, no trigger, at least under the coordinated framework. Therefore, no statutory authority. Therefore, they send letters out going, under our authority, we have no concerns about this. And suddenly people went, yeah. Okay. It was named the Breakthrough of the Year. And meanwhile, in contrast, in 1989, a very small research company was able to make transgenic salmon. 1989. When did they get approval? 2015. How many years is that? Why was it so hard for an animal, a transgenic animal, to get approval when it was, 1995 for crops? Say again?
Plants don't have feelings.
Sure they do. You
never heard of such a plant?
Okay. How many people here know a plant breeder? That's a great thing about Madison. How many people have ever heard of a fish breeder? Are there very many? Are there cultivated varieties of fish? I don't know about that, because that was one of the problems, is that the FDA was saying, there's cultivated varieties being brought up all the time through these huge plant breeding programs. We kind of grow fish at the fish hatchery and stuff, but there's not so much in the way of a route to comparison. And that's, I think, one of the major reasons why it took till then. Now, because of in part, and probably no small part, of the CRISPR thing, the Obama administration launched a review of the coordinated framework, the first review since 1992. And in September of 2016, the Obama administration issued their proposals to revise it. And on November 8th, Hillary Clinton was elected president. Holy cow. Okay, so when people woke up on November 9th or 10th or 11th, that's about as cataclysmic of a political change in our field as there is. The Obama administration had till noon on January 20th to do whatever they were going to do, so they released the final version of their proposed update to the coordinated framework on January 4th. And on January 19th, they were published in the Federal Register. And at noon on January 20th, President Trump was sworn in. And that afternoon, he issued an executive order freezing all proposed regulations, including the coordinated framework. So, is it in limbo? I don't know. We'll talk about that. The other thing that's pretty cool is not only was the aqua bounty approved, but they've got their first sale, and they grow these transgenic fish in Panama, and then they ship them up to buyers in Canada. Updates. So these are the 4 things I'd like to go into more detail on. What's the status of major transgenic crops worldwide? The transgenic salmon, a little bit more detail. The GMO label law from last year, and gene editing, and the Trump administration. Holy cow! How many millions of acres are there grown in the world of biotech crops? 457 million acres. Which countries are they grown in? USA, Brazil, Argentina, Canada. What are the major crops?
Corn, soybeans,
Canola and
rapeseed.
canola is rapeseed. Yeah. What's that?
I'm saying that was canola.
Yeah. I'll just go with those 3 because that's you're going to see what the others are, but those are the major ones. And cotton. I forgot about cotton. Okay. Is cotton a food plant? Let me ask you that again. Is cotton a food plant?
You're a bowling guy.
I make the bad jokes. You're most laughing. Does anybody in here use cottonseed oil? Absolutely. Is it a big feed plant? Yes. You are 2 blocks from a silo that is full of cotton seeds because it's used by the dairy cattle feeders on campus. And how many cattle are there on our campus? About 80, 83, 85. Okay. So do look at those numbers because what you're seeing is the biggest number of acres or hectares is actually in soybean, hence Brazil, Argentina, and the United States. Canola is the United States and Canada. And then maize. And what amazes me is how little of the percentage of maize is transgenic. I was surprised when I saw this. And that's because so much maize is grown in other places besides the United States, especially in Europe. And Europeans don't grow much in the way of GMOs. Now, what I would like you to do is think about what crops aren't there. So here's a list of the top crops in the world, United States, UK, South Africa, blah, 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 blah. And what you're going to see is, wow, outside those top 4 that I just showed you, there's all kinds of crops that are really, really important, wheat and rice in particular. And where's the transgenics? You can make transgenics, but you can't sell them. Let's go on to the fishy. Of these fish, which one is the genetically engineered one? Do you have any way to know? No. Just because one is bigger than the other, does that mean it's transgenic? No. Which one is going to get bigger at full size? This is what's so misleading about this photo.
About the same.
I don't know what gene. They're going to be the same, and here's why. The transgenic one gets to its full size faster than the non-transgenic one. Okay? But if you were to look at this picture, it would fit into the Frankenstein image of, wow, we are making monsters. We are transmogrifying food. And that's not the case. Okay, so like I said before, tough thing with breeding salmon is that there's no clear route through regulatory. I bought some transgenic fish for my daughter. They're called zebrafish. They were approved as pets. You can still go buy them today. These will not be labeled in Canada. They'll probably be labeled in the United States because of the new label law, but that's only because of the new label law. What about that new label law? Well, let's back up a little bit and talk about food labels in the United States. Food label has to do 2 things. It has to be truthful and not misleading. And the first time I heard that, I jumped up out of my seat and said, how could something be truthful and be misleading? I was young and dumb. Okay? So, for example, I buy orange juice that says GMO-free. And why is that, in my opinion, a misleading label? Because there are no GMO oranges in commerce.
Like fat-free hard candy.
like fat-free hard candy. Okay? Yeah. Okay. So, case study, BGH-free milk in 1994. Vermont, Wisconsin, and Illinois took 3 different approaches. Vermont said, must have a label, it's compulsory. Wisconsin said, oh, you can have a label if you want to. You got to use that great big long one that the FDA approved. This milk is from cows not treated with RBST. There's no significant difference been shown between milk from treated cows and untreated cows. Brevity. And Illinois said, no way, Jose. Any mention of this stuff, since there's no significant composition difference, is misleading. So those are 3 states, 3 different ways of looking at it. One with a compulsory label, the other had a permitted label, and the third one prohibited. There's this idea the consumer has a right to know. Do you have a right to know? Sure. Do you have a right to know the blood type you get? Yeah. Do you have a right to know where your blood comes from? If you get an infusion? Really? Let's talk about President Truman. Because at the end of World War II, how many different blood supplies did we have in the United States Army? 2. One for white folk. So that's an extreme example, but it's one that drives home. You may think you have a right to know, and yet maybe you do, maybe you don't. So let's look at easier stuff on right to know about food. Do you have a right to know everything about your food? I don't know. But I think under legal ideas, the strength of a right is largely determined by the remedy when denied. What's your remedy if you believe you have a right to know and the farmer won't give you the information, or the food seller won't give you the information? You have the right not to buy. Okay. Now, I have a friend by the name of Robert Streifer, who's a professor of philosophy here, and he's got out a very nice paper from 10 years ago, maybe a little longer, that makes a very cogent argument that if enough people have believed that they should have a right to know, that should suffice in a society to provide that right. And so that's a pretty cool thing. It's another interesting thing about working at a university. There's also the economic justice argument, which I first heard from 2 researchers over here in the School of Human Ecology. And the economic justice argument is this: all other things being equal, and not counting compulsory information that all foods have to provide, information about a food product should be considered a good. And under the economic justice argument, people who value a good should have to pay for it, so that people who do not value a good don't have to pay for it. And that's a yin and yang to the right to know versus the economic justice argument. And I already mentioned the idea of rights versus remedies. So that was before 2016. In the summer of 2016, the U.S. had, up to that time, had no specific federal statutes relating to biotech and food labels. Many states had either passed, Connecticut, Vermont, or were seriously considering them. And the only route that people had was to the national organic standards. It was an indirect option. Biotech varieties of crops cannot be certified organic. That was a huge loss for those of us in the biotech field, because when I started at agrogenetics, that was one of the big things we were hoping for, was that genetic engineered crops like corn that had a organic approved pesticide in them, like Bacillus thuringiensis Bt, would meet the organic standards. That's not how it worked out. And you still had this issue of unintended contamination by pollen drift or mixing of grain. So let's look at corn. Field corn is fungible. You grow field corn if it meets certain characteristics, doesn't matter what variety it is. What happens if you grow blue corn? What happens if you grow sweet corn? What happens if you grow popcorn? Those are all premium priced types of corn. Who's responsible for planting their corn far enough away from other people's corn? So this precedent is one that I would bring up 15 and 20 years ago, and in my estimation, it got exactly nowhere. So the burden of proof originally, the onus was originally on the premium priced grower. But the folks that grow organically contend, even though they're the premium priced grower of, say, organic sweet corn, the fungible folks that are growing BT corn, for example, should be the ones that bear any cost of contamination. So, July of last year, we get the first law. It gives 2 years to develop rules by the Secretary of Agriculture, but that'll be okay because it's going to be a Secretary of Agriculture appointed by President Clinton. And then November 8th happened, and we now have a Secretary of Ag by the name of Sonny Perdue. And that's different than Purdue University. I do like to point out that Purdue is the past participle in French for the word lost. So the label is very different than, say, the label that you would see in 1994 on Flavor Saver tomato, which was voluntarily labeled. It can be a text, or it can be a symbol, or it can even be a QR code. And they have 2 years to develop that, starting from last year, so they have 1 year to go. And who will decide what's acceptable? The Secretary of Agriculture and the Trump administration. And this is supported by large ag and food companies as a way to avoid state by state by state by state label laws. It's growing late, so I'm going to skip through this. Okay, now, woo. You've all heard of Frankenfood? Okay. One of the great devices ever invented. Mary Bissy Shelley, way to go, 1818, Frankenstein. But this is an example of vitalism. If you want to get better vision, and there's a dead eagle, what do you eat? It's eyes. That's what vitalism is. You want to get the vital component of that other organism into you. And that's how a lot of people would look at with transgenics. If you put a fish gene into tomato, what's it going to taste like? So the cool thing about gene editing is instead of having Frankenfood, you have deleting food. It's not what you add, it's what you take away. And I made this up when I was a speechwriter. I still believe it. But now it's a metaphor for gene editing. So there were other things before CRISPR, like RNA interference and zinc finger and talons, stuff that I could never follow. It was difficult, it was hard. They could do it on occasion, but whoa, along comes CRISPR, and now you can really turn on or turn off the expression of a gene that's already present in a plant by turning on or turning off, usually turning off another gene that's in the plant. What do plant breeders do? So look at that date, January 1st of 2015. This is the New York Times. Holy cow, if you edit the genes, you bypass the regulation. Because there aren't any foreign genes anymore. There's no trigger. So now what has been approved, at least by the USDA, slower browning apples. Not just Granny Smith, but other apples that are red and taste a little sweeter, not so tart, and you knock out some of the polyphenol oxidase enzymes that turn your apples brown. Same thing with mushrooms. The waxy trait for high amylopectin in corn. These are just some of the examples. So now we're going to revisit this coordinated framework, largely triggered by the CRISPR thing. Remember, it came into play under the Reagan administration, and was revised in 1992 under the first Bush administration. And who was vice president at the time? Dan Quayle. And who was in charge of this? Dan Quayle. And you, sir, are no John F. Kennedy. And it used those existing statutes. So we're going to update this thing. We're going to use the existing federal statutes. We're going to continue to use mostly those agencies. And supposedly it's going to be risk-based and therefore, at least in theory, based on the product and not the process. So we already went through some of this. While this is happening, in part because it was happening, the administration commissioned a National Academy report that came out last year. 2 faculty members from this university were on that academy. And they repeated what has been said by the National Academy for a long time. Regulation should be based on novel characteristics and hazards. All technologies present threats, therefore it should be the product and not the process that is regulated. That is not necessarily what the U.S. ends up doing, but that's what we say we should do. Holy cow. So for the first time in my memory at least, a National Academy of Science report started pointing out, wow, with editing, this becomes more difficult to say, how come we're regulating this and not plant breeding? So I'm wondering, are we going to come into an era of comparable due diligence, which has always been what many of us had hoped for. So as we have these molecular approaches, move away from moving new genes into crops and towards editing existing genes, what are the difference between the older and newer methods of plant breeding? Are they different or are they indistinguishable? So if they're more and more asymptotic to each other, any talk that I get to use the word asymptotic is a successful talk. How long are plant breeders going to be excused from regulation? And keep in mind, this is in the background that costs of DNA sequencing continue to drop, that speed's going up, the cost of mass spectroscopy to analyze the proteins and lipids and carbohydrates and all that other stuff also continues to increase in speed and decrease in cost. It'll be harder and harder to say we don't know what changes we're making because we can't know. So there's going to be a reckoning, I think, of available intelligence versus due diligence. Oh, excuse me, the impossible versus the inconvenient. So this will not be necessarily happy if this is where it ends up going with because the plant breeders have not had the same diligence required. So what's coming? I don't know. But there are plenty of ideas for thaw. Remember, right now we're in a freeze, but when the thaw comes, let's see what might change in the law, if not the statute. So Val Giddings is a guy that's been doing biotech and food policy for 30 years. He published this back in April, and these are his bullet points. And he's basically saying, whoa, these regulations should be based on process, not product. Excuse me, the other way around. Product, not process. There shouldn't be any process-based regulations from APHIS. The FDA, same way. The FDA should back off its regulating of gene-edited animals triggered solely by the gene editing. So that's different than the USDA's approach to crops. Same thing with the EPA. A little mention of the Fish and Wildlife. And then, one of my favorite things, if you ever want to get really excited about something, study the World Trade Organization and hold on to your seats, because glaciers move faster than WTO. Okay. So this is the math that a lot of conservative folks, I think the UK had the same thing. I think this actually is from, who was the woman prime minister? Margaret Thatcher, I think. I can't remember exactly right, but I think she had the 2-to-1 idea first. So I'd like to leave you with this idea, is it safe versus is it safe enough? That's been the crux of my work for 26 years, is to get people to go from asking, is it safe, to is it safe enough? Because science can help assess the risk, but politics draws the threshold of acceptance. I have the speed limit up there because 65 is legal and 66 is not, but there's no scientific reason necessarily for drawing the line at 65 and getting a ticket at 66, but you still have to draw the line somewhere. And with that, I went long, but I'd like to stop and take any questions. Thank you. And I hired Wynn to say, can we leave now and go to the bar? Yes, you can. Hey, it stopped at the ATM and it gave me money. I know. Questions?
So how do you prove something's safe? It's not dangerous.
Well, like I just said in my last slide, if anybody ever asks me, is it safe? I always try to reframe that as, I can't answer that. But I can say, is it safe enough? Because we have other things that we have already agreed are safe enough. And then you have this issue of comparability. Now, this one might be capricious, but it's already there. It's set by precedent or tradition or whatever you would like. So if you have a new technique or technology or whatever, how safe should it be? It shouldn't be less safe than the existing one. But if it has to be more safe, you have a huge opportunity lost here. Huge opportunity cost. So well, I shouldn't say most people, I've never asked them. One of the most important people in the world to me thinks that these thresholds ought to be comparable and that you should assess based on risk. And the other thing is, because I didn't include this, the whole idea of reassurance regulations. Some people think because something is new and people are squeamish about it, therefore we should put in regulations that will reassure the public. But and this is not my argument, it's from another person, but it's an argument I've been trying to make for 20 years once I heard it. Once you have reassurance regulations in there, whoa, can anybody tell that it's for reassurance and not because of risk? It sends the message that it's taboo and different. And so that's the problem, from my point of view, of having reassurance regulations. Even if you argue that, the risk is the same, but we need to have higher regulations, the perception is, therefore, almost immediately becomes they are more dangerous because the threshold is higher. Kai?
Yeah, how much does a role of media perceptions play in the general public's opinions of gene-edited foods?
How much did the role of media play? I don't know, but if I really want to change something, I'm going to be a cartoonist. Because one cartoon can capture and project one image much better than a bunch of words, with the possible exception of really exquisitely crafted words like Frankenfood, Terminator. Does everybody remember the picture of the bacteriologist in the moon suit when they're spraying frostband? What does that send to you? This stuff is dangerous, and we're spraying it on strawberries. So it's in the media, to me, it's the images that are the most compelling, the most long-lasting. They feed into the vitalism argument, even more so than a lot of words. So I guess that's how I'd say that.
Is golden rice going anywhere?
Is golden rice going anywhere? You didn't see it up there on the ones. So this golden rice is since 1999. It had a lot of problems, but I think the difficulty is even as late as 2014, they were still having experimental plots ripped up in the Philippines. So I don't know if it's going to go anywhere or not. Sherry Tandy-Mahardio gave a talk from this lectern 3 or 4 years ago about high vitamin A corn. And what is the color of high vitamin A corn? Orange or yellow. Okay. What is the color of rice? Well, a lot of it's white. And what is the color of high vitamin A rice? Yellow. So one of the things corn has going for it is it's already yellow unless you're growing white corn, but most people don't remember that most corn used to be white. If you ask a kid to draw an ear of corn, it'll be yellow, and even more brightly yellow with the high vitamin A corn. So I don't know where that'll go, but I still think the vitamin A deficiency is a great example, and this is what Ingo Petrikis argued. He's the guy that was the lead developer of golden rice. This is a moral failure when you have millions of people dying and people say, oh, you shouldn't use this. It's like, okay, because one of the things that people had said all along since 1985, this is only about profits and it's only about growing more corn when we don't need more corn. When are you going to give us something that's really important? So if you've heard my talk before, my other talk, I spent a lot of time on golden rice, but I haven't heard that it's any further along than it was 3 years ago. Other questions? Yes?
I have a question. I read a book this year called Wheat Belly. I don't know if you're familiar with it.
The book called Wheat Belly?
Yes.
Okay.
Written by William Davis, a cardiologist in Milwaukee. And he contends that bread, wheat, causes all sorts of illnesses and everything has been so genetically changed. So did you have a comment on that?
Well, I'm going to stop eating my brats with a bun.
I've already did that too.
Well, whoa. We share an idea. Here's something that's interesting was the whole celiac. Am I pronouncing it right? Is it celiac? Okay. Celiac is a mystery to me. I don't have it that I know of. Same thing with, is that the same thing as gluten tolerance? No. Intolerance? So celiac is different. So you have these two things that are much more prevalent now than 10 or 20 or 30 years ago. They both involve wheat and its close relatives. And imagine if transgenic wheat had been on the market. Who would get blamed for that? So, very interesting. This would be the first case in human history that a physician wrote a panacea book. And what's the French? What's the Latin word for bread?
Can you think of any clear mistakes that have been pulled off the market or beyond public pressure, just, oh, we don't want it, but I mean something that it got out there and it was like, oh my God, we really do have a problem with this stuff, we better pull it.
Yeah.
We discovered it down.
The two big ones that I know of are Starlink and the soybean that had the Brazil nut gene in it. Yeah. So Starlink was a mess, not because it was a problem. It was a mess because it was approved, but only for use in animal feed and industrial uses, not for humans. And the problem with that was, do we have two separate streams in the United States? And the answer is no. So you have an adulterant out there. You have somebody that mixes Starlink corn, which is legal only for use in feed and industrial use, and if some of it gets into human food use, you have a big problem, and that's what broke out in November of 2000.
Do you have a big problem, or is it just that it's not approved? In other words, was there something wrong with it that humans shouldn't be eating it?
No.
Or they just didn't know?
Correct.
And the error there was simply one of, well, it's not approved.
Correct.
It's not that it's bad. I mean, have you actually found anything that's come through the system that's proven to be bad at the far end that had to be pulled out because it was bad? It wasn't
Does anybody know one? Because the soybean with Brazil nut was pulled before it was approved.
Was that an allergy problem?
Yes, so that's a common denominator with both of those. The concern with Starlink was this modified BT might be an allergen. How do you test if something might be an allergen? That's an epistemological morass. Okay? It's a lot easier when you have something that's a known allergen, like the Brazil nut, because what can you do? You can go to the 1 in 200 or 1 in 2,000 or 1 in 20,000 people who are allergic to Brazil nut, and what do you ask them for? A donation of blood. And then you get their blood serum, and you can test whether that, without killing them, whether the protein that's being expressed is the one that causes the allergenicity. So epistemologically, you have two very different things. One where you have a known allergen and you've got a reagent, and the other where you have a possible one but you don't have a reagent. So how do you ever prove that this is not an allergen? And that's why it's an epistemological morass.
That is a good question, though. Has anything actually gotten out that was demonstrably dangerous to people?
Does anybody know of one? So the big one right now that people are, what's that?
Because you're saying there's some things and maybe they're bad. We don't know.
Okay.
'Cause the way
I will say this, as far as I know, no biotech crop has ever caused anything so much as a headache, except for the political headache and the financial headache. If somebody knows of something, let me know. But that report that just came out said we don't have any examples of that, which doesn't mean things haven't been culled as they go through. But I can't name for you something. And if there is, let me know. Because it makes me look stupid if there is and I don't know it. But hey, I look in the mirror every day. Win.
I guess the issue of designing crops to work with particular herbicides.
Yep.
Because they are running into problems with that, where they've got a crop that they've engineered to run with an herbicide that hasn't been approved for use yet. Some people are illegally growing it in their fields. It's drifting in their spraying this crop with it, and it's not illegal to grow the crop, but it's illegal to use pesticide. Their neighbors don't have the resistant crop, and their fields are being damaged by spraying.
So that's pretty close. It is legal up until about a month ago because they put an emergency ban on the dicamba. So starting in 1996, one of the big things that came out was Roundup Ready or glyphosate resistant soybeans and corn and cotton. And why would you want that? Because Roundup kills everything unless you're resistant to it. And how do you get to be resistant to it? Somebody gives you a gene, like Monsanto. Okay? And then you spray, and you have fields that are perfect. For a few years. And what started appearing was this pigweed, big one called Palmer amaranth was the first one that I think certainly the one foremost in my mind, especially in cotton, especially in the South, especially in Arkansas, some other places. So now you're paying extra money for the seed, and you're overspraying your crop with Roundup, and it gets all the weeds but the one that grows to be 8 feet high, and screws up your combine and has hundreds and hundreds of thousands of seeds. So when it goes through the combine, what are you doing? Whoa, this is good. Okay, so I heard somebody say, "Oh, this is evolution," and I said, "No, I don't think this is evolution. This is what Darwin would call artificial selection. This is not natural selection. This is artificial selection because we're selecting for it, not nature." The whole idea behind letting natural selection happen is why you have 80% of your corn seed with BT and 20% without, so that you let natural selection fight the artificial selection. What's the solution? go back to all the other ways we used to farm in 1995. What happens to no-till farming? Or you get another one like Liberty Link and you use Liberty herbicide, or you use dicamba-resistant crops. And the problem with dicamba is, apparently, it volatilizes. In other words, you spray it, and then comes a hot sun, and it'll lift off the leaves and drift over here to a field that doesn't have dicamba resistance. So this is a major thing going on in Arkansas in particular right now, of trying to say, are the formulations the ones that have been approved, or are people using old formulations of dicamba that are formulated in a way that really prone to this volatilization and drift? And so last week, Monsanto went to court to fight the Arkansas Plant Board over the ban. The EPA has issued a ruling on dicamba, so that's the one that's happening right now. The reason I'm in a dilemma about that because it's not so much the genetic engineering aspect, it's the resistance. And the overuse or use of a trait. For those of you who remember 1970, what happened to the corn? We had the corn leaf blight, and it was the worst corn disease year in my lifetime. And how did that happen? Texas male cytoplasm. Blame the Texans if you can, especially the males. Okay, and so there were all these strains of or lines of hybrid corn out there, and you didn't have to detassel because they had Texas male cytoplasm. The problem is the yeast, not the yeast, the fungus that caused the blight targeted that gene product. So you can get the same type of vulnerability if you have widespread use of even a single gene from traditional plant breeding. What are the 2.5 happiest words in the English language? All right. So thank you very much for coming. I'm going to